Good morning, everyone. We are at the genome.ucc.edu homepage, and today we are going to talk about in silico PCR. And if you look at the description of in silico PCR, it says rapidly align PCR primer pairs to the genome, which means you have a primer pair that will serve as input, and you're looking at where these primer pairs go and align on the genome. Right? This will serve you two purposes. One, if you've forgotten the target of your primer pairs, you would get to know what exactly is the region of the genome that these primers are supposed to amplify. And second, it will also tell you if there are other regions that are not targeted, but will get amplified because the primers have other complementary binding sites as well. So these are the two purposes of in silico PCR. It's kind of a quality check on the primers that you've designed. So let's go to in silico PCR now. So we are now at the home page of UCSC in silico PCR. And if you see the top panel, there is input for the genome. So by default, the human genome is loaded. Then there is also an input for the assembly. And by default, the GRCH38 or Genome Reference Consortium 38 assembly of the humans is loaded here. Then by default, the target given is genome assembly. And then you have the option for submitting your forward primer and the reverse primer sequence. Then you have an option of maximum product size, and by default, this is 4,000, but you can modify it in case you know that your product is going to be a little longer than this. Then there is an option for minimum perfect match, which is basically to say that a minimum number of 15 nucleotides must match the target at the 3' end. Then you also have an option for minimum good match, which is again by default 15. And this basically represents the number of bases on 3' end of primers where at least 2 out of 3 bases match. And then you have a last option of flip reverse primer. So what is flip reverse primer? Basically, if you are taking the reverse primer sequence from the top strand itself, then obviously it is understood that the reverse primer sequence would be a reverse complement of the end of the top strand. So you could do that here by checking this square, right? So now let's look at the utility of the in silico PCR. So let's say what I have is a primer pair and I've forgotten what is the region of the human genome that they amplify. So here is my forward primer sequence and here is my reverse primer sequence. So I want to check one, what is the target region that they amplify? Second, also I want to check whether these primers are specific and go and complementarily bind only to one specific region in the human genome and not to multiple other regions. So I copy this forward primer sequence and paste it in the forward primer section here. So I say paste. Likewise, I copy the reverse primer sequence from here and paste it on the reverse primer sequence option, right? So here we are, and I paste it here again. And also because this is a primer that I know is representing the reverse primer in the correct orientation, I will not flip the reverse primer sequence, right? And then because I'm not sure what is the actual length of the target that they're amplifying, I may increase this range to, let's say, 7,000, 10,000, whatever. So let's say we do 7,000, right? And then, of course, you can click on submit. So here you are, you can submit now and see what is the exact region in the human genome that these primers are going to amplify, and whether there are other regions of the human genome that may be amplified with the primer set. So we say submit here. And there you are. If you see, there is only one hit, which means only one region of the genome is going to be targeted by these primers. And the coordinates of the amplicon are given here. So there's going to be chromosome 12 from position 6534478 to 6538457. The total amplicon size that you're going to get is 3980 base pair. This is the forward primer sequence. This is your reverse primer sequence. And then the amplicon, the nucleotides that come in between these two primers are given here. And you can see this is the whole length here. The primer sequence is given in capitals, and the rest of the sequence that gets amplified is given in small letters. And here you can see very clearly there is only one amplicon that you obtain, which means that these primers are going to be specific. Second, you also want to know what is the region that is getting amplified. So for that, you can click here, and this will take you to the actual annotation for the region, right? So when you zoom down here in the browser page, you see this is going to amplify the GAP-DH gene. So this is the start and end of the sequence that is getting amplified, and this region is annotated as the GAP-DH gene or glyceraldehyde phosphate dehydrogenase gene, right? The chromosomal location, again, it is the same as what we had seen in the annotation. and the amplicon length will be 3980 base pairs. So this is the utility of in silico PCR. It can tell you what is the target for a particular primer pair. And second, what it can also tell you is whether your primer pairs can also secondarily anneal to another site that is similar. In this case, there is none. So therefore, you get only one PCR product. That is your targeted gene, the GAPDH gene. Thank you very much. And I'll see you again with another practical very soon.